I hope you took some time to think about the facts that were known about DNA and what its structure might be, setting aside what you probably already knew about the structure. This is a picture of Watson and Crick showing a model of their structure. Remember the facts available. DNA transforms cells, the base equivalency, adenine equals thymine, cytosine equals guanine. This had to be accounted for in their structure. There's a 3' to 5' phosphodiester linkage. The 3' carbon of one sugar connected through phosphate to the 5' carbon of the next. The helices run with phosphates along the outside and bases along the inside. This is a flattened version of the Watson-Crick helix on the left and a model on the right. First, notice that the linkage between the two bases is between the 3' of one sugar and the 5' of the next sugar, and that the linkages run in opposite directions on the two strands. So these are anti-parallel strands. If you look at the ends, they each run 5' to 3' because the phosphate on the 5' end is available and the 3' end has the next hydroxyl. For the two strands, the bonds run in opposite directions, so the orientation of the strands is opposite directions. This is another view of the structure, and what you'll see is that pyrimidines pair with purines. T is paired with A, C is paired with G. Non-covalent hydrogen bonds form between the bases to make base pairs. And this pairing is the part of the structure that accounts for the base equivalency established by Chargaff. There are two grooves in this structure. The major groove is wide and the minor groove is narrow. And you can see that even better in the space-filling model on the right. That comparison between these, the wideness of the major groove and the narrowness of the minor groove. These grooves allow access to read the bases and therefore also the base pairs, because if you can read one base, based on those base pairing rules, T pairs with A, C pairs with G, you can tell what one base would be paired with. These grooves are very important because they provide a recognition surface to read the sequence of DNA, even though those bases are sequestered inside the sugar phosphate backbone, which is very polar. That backbone of each strand is held together by covalent phosphodiester bonds, the alternating sugar phosphate backbone, and thus the surface of DNA itself is highly negatively charged. The two strands come together through non-covalent hydrogen bonds that hold these bases together at the center of the structure. There are also apolar stacking interactions indicated by the arrows that stabilize the structure. That is, that there are higher energy bonds holding the sugar phosphate linkages of the backbone compared to the non-covalent interactions between the base pairs. The helix itself is right-handed. That is, if you hold your right hand with your thumb pointing upwards, your fingers will wrap around the helix in the direction that the strands wrap around each other. So DNA is a right-handed helix with hydrogen bonds between the two strands and covalent phosphodiester bonds creating the polymeric structure. DNA has about 10 base pairs per turn, with 3.4 nanometer rise or distance per turn of the helix, 0.34 nanometers per base. The diameter is 2 nanometers, so you can deduce something about this molecule. There are many more bases in most DNA than is shown here, and the length will change as bases are added, but the diameter is constant, 2 nanometers. Here's another view of DNA where you can see the sugar phosphate backbone on the outside, the bases stacked on the inside to stabilize the structure, and you can see the hydrogen bonded pairs listed at the center. The base pairing, G with C, A with T, keeps the diameter uniform as you add length to the structure, because a purine is pairing with a pyrimidine. Also, you can read the sequence from either strand, because if you know the sequence of one strand, you know the partners in the other strand. The implications of this pairing were obvious to Watson and Crick, and you can read their one-page report. You will see that they were quick to imply that this suggests how DNA might be copied. Interestingly, there are multiple forms of DNA. The B-form structure, which is the Watson and Crick structure that we've been looking at, but there is also A-form and Z-form. The A-form occurs under very dry conditions, low humidity, and you'll notice that it has almost no minor groove. Take a minute and notice what's different about the Z-form. Look at the arrows that follow the backbone in these images. The Z-form is left-handed DNA, and it too has almost no minor groove. B-form is the primary form found in cells, but Z-form can also be found in cells, and its formation is favored by certain sequences of bases. We'll focus on the B-form because it's the majority of DNA in cells. Think about these reflection questions. What types of bonds provide the major energy in stabilizing the interaction of the two strands in double-stranded DNA? And what does parallel stacking of the base pairs suggest? What are the implications of this structure of DNA? Think of at least two, just from considering the structure, and identify an experiment that these implications would suggest is worth pursuing.